Alright, in this video we're going to be talking a little bit about the photoreceptors or the visual pigments in the eye, as well as how they are affected with light. So basically it's really important to think about the specialized parts of the eye which can detect light. So in rods we call this opsin. This is the protein, and it's connected to another part, which is the retinal. Now, the retinal is the actual part that can detect. This is actually the part which can actually detect the light, and the opsin is actually the part that can actually like hold it. It's more of the protein. But this is actually not just called the retina. It's called the 11-cis retinal. And it actually comes from, it's actually from a vitamin A derivative. So it's actually broken down in that way. And interestingly, cones have 3 different kinds of opsins, right? And sorry, rods, so cones have 3 different types of opsin, whereas rods only have 1. So basically, this is why cones are able to give us a much more sort of like diverse amount of acuity, whereas rods kind of do one thing quite well, and their adaptation rates are quite consistent. So we'll talk about that a little bit later. But basically, let's have a think about how this process works. How do we go from 1 photon of light into literally thousands or hundreds of thousands of movements of different ions that allow the rest of the the organs and other responses of the body to work? And the answer is through a G protein system. Now, if you haven't heard of a G protein system, I'm not going to be able to go into it in a lot of detail in this video, but it's just really important to think of it as an amplitude system. It's a system in which one single signal is actually amplified through different messengers throughout the process and gets bigger and bigger and bigger. So in this case, what we're talking about is a system that basically is based on a G protein called transducin. Now write that here. Now transducin is connected or associated with these molecules. So we just talked about rhodopsin, which is this whole molecule. So we talked about the opsin part and the retinal part. Together, this whole thing is called rhodopsin. And so what happens is the rhodopsin molecule, which is what we've talked about here, this whole thing here is connected to a G protein called, so we've got rhodopsin, we got transducin, and then finally we've got something called PDE, which is actually phosphodiesterase, which you probably don't need to remember, but just in case, phosphodiesterase. And the way this process basically works is that light will come in, will activate rhodopsin, which will turn on this G protein, which then will move and conform shape, and then PDE will go about making some changes to some molecules. Now, the star of the show of these molecules is a molecule called cGMP. Now, the one thing I want you to remember about cGMP is it basically keeps ion channels open, okay? This will become really important in a second. In this case, it keeps ion channels open. You have cGMP, ion channels open. If you don't, they close. That's the main point. So let's put this in a little bit of context about what this looks like. So imagine in these rods, we have two membranes, and I'm just going to draw these membranes quite rough. And if you've seen a proper membrane, you should have by now. They'll be very, very different to this, but this is just a sort of rough idea of what they look like. And this is basically the outer rod membrane, and here you basically have the different ion channels. Now, what will happen here is because there is a higher density of sodium outside the cell, these will come through just purely through facilitated diffusion, will come into the cell via these different channels. And then what ends up happening is that you get a increase in the intracellular rate concentration of sodium. Now, the interesting thing here is that the cell doesn't necessarily always want to have that, and so what it ends up doing is there's an Na ATPase pump, which basically pumps out all of that excess sodium. So it's kind of like, it seems sort of like a redundant process, right? Like you leave these channels open, the sodium leaks in, then it's immediately pumped out. Why do we do that? Well, it also is important to start thinking about it from the context of what ends up happening when you close these. So the other thing to think about is, so in all of these situations, we've got cGMP. It's sort of holding everything together, and it's keeping the channels open, right? However, on the other side of this, we've got on the disc membrane, which are over here, the disc membrane. We've got our rhodopsin molecule here. Then remember, we've got our G protein, and that's called transducin, which we'll draw here, transducin. Then we've got another particle, which is called PDE. And PDE is the enzyme that will go around and change things. So I'll run you through how that works. So everything's running fine. You've got these cGMP molecules holding these ion channels here open. Sodium's flowing into the cell, then being pumped out of the cell. Everything's going well. All of these at this stage are inactive. Nothing's happening. But then we get light. Now what happens when light happens is the light rays shoot in to the cell. They hit this rhodopsin molecule here in yellow, and it becomes activated. So it's inactivated. All of a sudden it turns on. And in turn, it moves the G protein called transducin. Transducin then activates that enzyme called PDE. And what PDE does is it takes cGMP in the cell and it breaks it down into GMP. Now, what will then happen is all of these places we have cyclic GMP holding these ion channels together. That's gone now. And what then happens is that all of these are now blocked. And because there's such a shortage of cyclic GMP, that blockage causes hyperpolarization. Now, just to recap, hyperpolarization is when the cell becomes more negative. And you're probably wondering, okay, well, it makes sense that you can basically have these Na cells, if they don't come in, it wouldn't get more, it wouldn't increase. Like if I'm not adding more positive charge, it's not going to go up anymore. But how does, how does this sort of work with how does it become so negative? How does the cell become negative? And the answer to that is twofold. Because of these pumps that are pumping sodium out of the cell, which we talked about before, which are the ATP, the Na ATPase pumps. They're basically taking Na that's in the cell and pumping it out, in combination with the fact that there is a leakage of potassium, which is higher here. There's potassium leak channels, which all of that ends up out as well. That causes the cell to go into hyperpolarization, meaning that the resting membrane potential goes lower. And it kind of looks like that. So if you think about light, the signal for light is different to other sort of typical neurons that you might see, where it will have to, in a neuron, you'll have to hit a threshold by increasing the positive charge in the cell, and then that sets off an action potential. In these vision cells, it's the exact opposite. We need to have a lower amount of light, of charge in the cell. The intracellular membrane has to become negative relative to the outside, and not just more negative, but really negative. It has to be hyperpolarized. And in that situation, that's enough to set off the process and start generating an action potential which goes to the brain. So just to sum this up, light activates something called these rhodopsin molecules here. Rhodopsin molecules turn on transducin, which is the G protein, which activates PDE, which then takes cGMP and converts it to GMP. And because we no longer have our cGMP to open these ion channels, then all of a sudden it becomes more negative inside and we hyperpolarize. However, when there's no light, we still have these cGMP molecules and they keep those channels open. And everything keeps running normally. So the interesting thing is that there is something called a dark current. And if you ever ask what a dark current is, the dark current really refers to the fact that when there's no light, stuff is still happening. We talked about that a little bit before. When you basically take out when you still have these cGMP molecules, you're still going to have Na flowing into the cell, right? And you're still going to have a little bit of potassium leaking in and out. And you're still going to have the NAAPTase pumping things in and out. That whole process is still happening regardless of whether there's light or not light at that level of the outer membrane. So this is what we refer to as the dark current or the standing current. And it's a flow through the receptors even in the absence of stimulus, and it's continuously happening even without light. Now, like I said, when light occurs, it actually goes and activates a huge amount it's really important to understand that one photon of light here that impacts and by the way, there's more than one, but activates a rhodopsin molecule, that has a huge impact because that will go and activate thousands of cyclic GMP phosphodiesterases, which is that PDE molecule we've talked about. That will then cause hundreds of different channels to close. That will then cause, literally hundreds of thousands, not millions of sodium ions to now start hyperpolarizing the molecule. And then that will cause a huge voltage change between the cell and outside the cell. And that voltage change is actually what determines the action potential to the brain. Now, it's really also important to think about the level of intensity of light. And so we know that when light happens, we should, like let's say the light happens here, we have a flash of light. What should happen to the cell is something like this. Normal, normal, normal. Then all of a sudden those channels close. We have a dip. Then over time it restores back to normal. that is an RP. You'll have, based on that, you'll be able to generate an action potential, I should say. Or in this case, it's called a receptor potential because it's actually going across the membrane to be graded. That receptor potential can become bigger and bigger and bigger depending on how strong the light is. So it can become and this is why it's a little bit different to action potentials, in that an action potential is an all or nothing response, and they happen a little bit later on. But the response, that level of hyperpolarization, it can be a little bit hyperpolarized, or it can be very, very, very hyperpolarized, depending on the intensity. And that is managed by a few different things. So the first thing is increasing the light intensity turns on more rhodopsin molecules. It makes sense. If you have a lot more light, a lot more of those G protein systems are going to be turned on. Then, of course, you're going to have more breakdown of GMP. If you have more systems turned on, that cGMP is going to get broken down into GMP. Then, because of that, you're going to have more closure of those membrane ion channels, which prevent Na coming in. And so it's really a graded response, isn't it? If you have a little bit of light, you may only become a little bit negative. If you have a huge amount of light, you get that big hyperpolarization, and those contribute to the action potential. Okay, let me ask you a question. What is going to happen if we have constant light? Based on what I've told you so far, you know that cyclic GMP, which keeps the ion channels open, is going to get broken down into GMP, and then what's going to happen is all those ion channels are going to stay closed, and we're going to get hyperpolarization. But we also know, based on that image I showed you before, that it does eventually return back to normal, and it needs to, so it can show additional light. And then how do we make sure that not only that happens, but we don't just use up all of the cyclic GMP with one bright light? So for instance, if I just show you like a huge light shining into your eyes, how do I make sure all of the cyclic GMP doesn't instantly get used up? So the way this works is by thinking about an enzyme which is called guanylate cyclase. And guanylate cyclase does the exact opposite of the process we were talking about before. So instead of breaking down cGMP into GMP, it converts GMP, make that G a little bit clearer, converts GMP into cyclic GMP. Now that process is really important because we need to restore the GMP levels so we can open up the ion channels again. Now here's where it gets interesting. At night, or when there's darkness, the ion channels are open because we already have cGMP. Now this is the really amazing adaptive system which balances this. So what happens is sodium sneaks in, because there's a concentration gradient out here of sodium. This is a sodium ions. So it's the main thing they'll sneak in. However, you'll also get calcium sneaking in as well. Not as much, but some calcium coming in. Now this calcium will basically impair to some degree the ability of, we'll just call it GC, which is the guanylate cyclase. It will impair GC from being able to maximally produce cyclic GMP. And that makes sense, doesn't it? Because If we've already got a lot of it in the cell, we don't want to have a huge amount and stockpile it, so in the darkness all of a sudden we have an excessive amount of this hormone, this enzyme. However, when cGMP is removed and there's less of it, it also means that these channels are going to be closed. Calcium cannot no longer get into the cell, and that impairment on cGMP is removed, production is removed. So when we need it most, cGMP levels are increased maximally. So what that basically means is that you have this perfect system, or near perfect system, to ensure that you're always producing the right amount of cyclic GMP when you need it, and breaking it down when you need it as well. So to summarize, basically, guanylate cyclase is the enzyme which will take GMP and convert it back into cyclic GMP. It does this in darkness at an impaired rate because the calcium is going to impair the cyclic GMP. However, when there is lots of light and these channels are closed and we do need more cyclic GMP to open them back up to sort of rebalance that polarization in the cell, in that case, that calcium is no longer there because it's pumped out by, I should mention, by calcium pumps. Just like we have sodium potassium pumps, we also have calcium ATPase pumps, which pump them out. And what you end up having is a rise in cyclic GMP levels, and these channels open back up all over again. So it is a really good system to be able to balance between that sort of levels. So that is basically a summary of what that looks like. And if I was to draw it, what would happen is, let's imagine we're sitting in the dark. Then all of a sudden, I get a bright light. It stays at that level. What will end up happening is that my resting membrane, we've already talked about this, will be about -40 MV. But what do we expect to the cones? Well, we know that as soon as there's a lot of bright light, rhodopsin is going to activate the G protein transducin, which is going to activate the process which will break down cyclic GMP, which will close the channels. Those channels will be closed, so we'll have hyperpolarization because of the leaking out of potassium. And what will end up happening is that we'll get hyperpolarization down here. But we also know that that process is restored and it comes back up here because of our friend CG, which is called guanylate cyclase, GC I should say. And that, because there is no calcium impairing it because the channels are closed, it will actually at a more rapid rate convert GMP to cyclic GMP much faster and restore the cell back to its normal resting membrane potential. Hope that makes sense. If you have any further questions, please post them in the comments. In the future videos, we'll talk a little bit more about adaptation and how that can impact the eye.